post. Do you have the schedule?
Not if that's an extra copy. I have it on my phone. But yeah.
Here's a copy. I'm meeting William later at that lake room.
Oh, yes.
Can you walk with him and Mark to the restaurant? I'll be there at 5.
Yeah, we have like.
So here's a paper copy.
Thank you. Oh, I didn't even see this. I got to hear you talk about your research. That's exciting. Great.
Yeah. All right. Great. Looking forward to your talk.
Yeah. Thank you so much.
See?
Yeah. Do you want me to give you a question?
Yeah, absolutely. I heard Valentina got her neuro.
Nice, great.
Oh, yeah.
Hopefully she'll come back.
Where is she now?
In Italy on sabbatical.
Oh, okay. Which university? Rome?
Yeah.
Oh, she's in Turin. I'm not sure. Yeah, I think she's there.
Okay. Do you have a mic?
I do. Let's see if it's live.
The battery's good.
Yep, that's it.
Basically just like the first one.
Testing. Testing.
Yeah. There's a spot
Is the live stream going?
Good. Thank you, everyone, for coming today. It's my pleasure to introduce Dr. William Hwang. He's a professor at Harvard Medical School and also an associate member at the Broad. He has an impressive array of scientific degrees spanning biomedical engineering, electrical and computer engineering, physics, biophysics, and chemistry, as well as an MD. And as I was looking through your resume, I also noticed that when you were at Duke, you founded a nonprofit called United InnoWorks Academy, which is a nonprofit for providing scientific education and engagement to middle school students, and actually has a chapter here at the U as well as many different universities across the country now. So he's today going to be talking to us about work that he's done in pancreatic cancer using a variety of omics-based and functional genomics approaches. Thank you.
Thank you so much for that way too kind introduction, and to Abby and Nelson and everyone else in the GSI for inviting me here. I actually just officially started my lab at Mass General Harvard Medical School last July. So it's exciting to share some of this. A lot of what I'm going to show today are not complete stories. I'm going to start with something that we did complete last year as kind of a introduction, but a lot of things that we're working on now as our team is growing, and I think an exciting time for questions in kind of tumor microenvironment of some of these very prevalent and deadly cancers like pancreatic cancer, with the new tools that we have available and are developing. So, any feedback later today or in the questions or potential collaborations would be of great interest to me. So before I start, I just wanted to acknowledge the people who have really been there every step of the way. All the work that I'm showing obviously was not done by me alone. This is our lab retreat at Lake House in New Hampshire this year. And this was, we were celebrating Jimmy's birthday for that, and then this was us at Putchak not that long ago. So the team's really been very awesome, and I feel very fortunate to get to work with them. This slide I put up here because, as with I think many of the labs here, nowadays there's this kind of interesting pairing that you have to think about when you look at model systems, technology, what questions you're asking. And I think hopefully today in the 3 stories or themes that I'm going to share on the work we're doing, you'll see kind of that thought process and why we picked the various elements that we did. So as a lab, as Dylan mentioned, I'm a physician scientist in the GI radiation oncology department at MGH. So I see patients for about 10% of my time, but I definitely think about them a lot more than that. And I think what we do in the lab is very much guided by the questions that arose from taking care of patients. And how I even got into the cancer neuroscience field, which will be the last story that I share, was very much based on taking care of patients as a medical school. So I'll share that story when I get to that part. But of course, with patient specimens, we're limited in what we're able to ask, especially mechanistic questions. So we also work with genetically engineered mouse models, as well as tumoroid and co-culture models, where we can really ask these mechanistic questions that we're interested in. So our lab, has focused on different aspects of these various techniques, from kind of the high-plex profiling techniques to more functional experiments, a lot more recently with live cell multiplex imaging. And I'll show you a little bit of an overview of a lot of these things that we're working on now. All right, so for the first, so here's an outline of what I'm going to talk about today. So the first story is mostly stuff that we published in a recent Nature Genetics paper, and then kind of moving beyond that a little bit into how we're trying to improve and further drill down on some of those results. And it's really asking the question in pancreatic cancer as the exemplar, but really I think these principles apply to other cancers and diseases as well. It's how can we understand therapeutic resistance, and at the single cell level, and also spatially, the context of the co-conspirators in the tumor microenvironment that play a very important role in the outcome of these tumors. And we are particularly interested with spatial techniques and looking at mechanisms of resistance that arise from cell-cell interactions. So not a purely cancer intrinsic or extrinsic mechanism, but really at that interface. And then the second part of my talk is going to look at some of these resistant cell states that we identified in the first part, and drill down on trying to identify what are the key regulators, transcription factors that drive those states. What is the context dependence? So what do these states look like in the primary tumor versus a metastasis, for example? And then, again, with kind of the translational outlook, what are the therapeutic vulnerabilities that may be unique to these resistant cell states? And then for the last part, I'm going to talk about this, I think, somewhat neglected component of the tumor microenvironment until very recently. That's the nervous system. And this isn't just central nervous system, which there's been a lot of really seminal work done by the likes of Michel Mange and Frank Winkler in gliomas and central nervous system tumors and those interactions. But the peripheral nervous system, for many tumors, including pancreatic cancer, head and neck cancer, colorectal, breast cancer, there is quite a bit more interactions with the nervous system than I think we appreciated previously. So understanding how that works and how that leads to pro-tumor phenomena, I think, is very important. Okay. So I think, for most of you here, if you study cancer, you know that pancreatic cancer, this was very motivating for me, is one of the most deadly solid tumors. So these are now slightly outdated. This is from last year, but the numbers are unfortunately very much the same despite the research pouring into this field. The 5-year overall survival still hovers around 10%. And various projections have pancreatic cancer being the number 2 or number 3 highest cancer-related killer by 2030. And various efforts to really try to understand how we can be better at the way we stratify our patients, started with genetic profiling, but other than the 1% or so of patients that have DNA damage repair mutations such as BRCA, mutants or mismatch repair deficiencies that respond to immunotherapy, by and large, those efforts to stratify patients by genomic alterations have not been very fruitful in terms of precision oncology. So about 15 years ago now, there have been multiple efforts kind of arising looking at cell state. So this started out with bulk RNA sequencing, as well as microarray technology. And these 3 kind of seminal studies looking at RNA level information really identified this kind of binary classification for PDAC that really has persisted to today. And that's the so-called classical subtype versus the basal-like subtype. And these subtypes are somewhat useful. So you can see here from this survival curve, they're prognostic. The purple curve these are bulk RNA-seq determined classical tumors tend to survive longer than the orange basal-like tumors. And they're also predictive of treatment response to multi-agent chemotherapy. So this is a waterfall plot of metastatic pancreatic cancer patients receiving mostly FOLFIRINOX. A few of the patients received Gemabraxine, that's what the arrows show. But you can see from the waterfall plot that the orange basal-like tumors respond overall less well to multi-agent chemotherapy than classical. And there's some nuance based on regimen that I won't get into. But there are many limitations, of course, from these early studies that really did establish the field. But most of these studies really focused on untreated primary tumors. There was some examination of metastatic samples, but pretty minimal. And then as the field has really evolved, we're noticing now that even with patients that can receive an upfront resection, which is typically a Whipple procedure if it's at the head of the pancreas, or a distal pancreatectomy, splenectomy if it's at the tail of the pancreas, those patients often benefit from neoadjuvant chemotherapy, and there's a lot of debate about the role of radiotherapy in this disease with mixed data. But certainly upfront chemotherapy seems to be beneficial for various reasons. And so it really increases the importance of understanding what do these tumors look like after they've been subjected to cytotoxic therapy, right? Because most of our clinical trials, they're all second line or third line. Patients have already seen at least chemotherapy. So having these states defined in the treatment naive setting is of a little bit more limited utility then. So this is data that we recently published, and on various slides I'll have pictures of some of the key contributors to these studies. What we did was, in collaboration with some of the MGH surgeons like Carlos Fernandez and the pathologists, we went into this frozen biobank that we had been accruing over a couple decades, and we performed an optimized single nucleus RNA sequencing approach on about 43 tumor samples. Some were untreated at the time of resection, and some had received chemotherapy, radiotherapy, and or losartan, which is thought to potentially have an effect in this setting as a TGF-beta modulator. We also had some patients here listed under other treatment who received various types of immunotherapy. It wasn't a large enough group to really do subset analyses on them, but the data is there. And we also did some matched spatial analysis with multiplex ion beam imaging, as well as NanoString digital spatial profiling at the whole transcriptome level, which I'll describe later. So this is a figure from the paper where we basically have a UMAP showing the cell annotations for the combined, all the tumor samples combined. And you can see that, using subclustering, we're able to delineate many of the expected cell types and subtypes that you would expect in these tumors. But most relevant to my talk today is when we were able to separate out the various cell types. For example, the malignant cells, we also did this for the fibroblasts. But for the malignant cells, we performed consensus non-negative matrix factorization to really try to take a de novo look at gene expression programs that were common and shared across multiple patients, that weren't patient-specific. And when we did that, we did various rounds of annotation. We compared these with gene set enrichment analysis, other various analyses. We were able to recover most confidently 7 gene expression programs, or cell states, if you will, that were recapitulated across multiple patients in our cohort. And some of these we already knew about from the bulk data. So classical is a very strong program that fell out of this analysis. The so-called basal-like, for us, we were able to resolve it into kind of 3 distinct subprograms that we call basaloid, squamoid, and mesenchymal, which kind of makes sense. If you look at the bulk literature, various studies call that overall program quasi-mesenchymal or squamous in the Bailey paper, or basal-like. I think with our increased single cell resolution, we're able to tease out some of the finer differences between those programs that were grouped together in bulk. We also saw a hint, it wasn't very prevalent, as you can see from these UMAPs, of malignant cells that had high copy number inferred copy number aberrations, but also expressed gene expression programs that looked like acinar cells or neuroendocrine cells. And that somewhat addressed, I think, a long-standing debate in the field of whether or not malignant cells could express these programs, or if this was just stromal contamination from the bulk studies. And we seem to see that at least a subset of cancer cells are able to express these physiologically normal programs, or some variant of them. Most interesting to us, and something that we hadn't seen a precedent for in the pancreatic cancer literature, was this so-called neural-like progenitor program, or NERP for short. And I'll spend some time talking about that. So here are some of the top GO terms from the MSIG-DB database for this program. And you can see hopefully why we named it what we named it. So neurogenesis, neuron differentiation, stem-like module, a bunch of hepatocyte nuclear factors that suggests, endoderm development. And then when we compared this program with the 1,500 or so brain-enhanced genes in the Human Protein Atlas, we saw that of the 7 programs, this one was uniquely enriched in brain-enhanced proteins. And in this heat map, you can see 4 of these proteins. Some of them, like Norexin-3, were not known to be expressed outside of the central nervous system at all, right? Down here, this is adrenal gland, GI tract, seminal vesicle, all other tissues. So what we did was we took some of our tissue samples and we stained them for pancytokeratin so we could identify the malignant epithelial compartment. And then we co-stained them for one of these brain-specific proteins, Norexin-3. And surprisingly, we saw that while most pan-CK positive glands didn't express any Norexin-3, some actually heterogeneously co-expressed Norexin-3. And this was surprising because prior to this, this protein was not known to be expressed outside the central nervous system. Then we wanted to look at our data. So one big caveat of this data is that it's not matched pre and post treatment. We're now generating data on various clinical protocols that are. But we started this study in 2018, looking at some of these samples, and some of these are historic samples. So definitely no matched data was easily available. But with the relatively large numbers that we had at the time, we were able to separate, at least for the 3 main treatment groups, patients into the untreated category, chemoradiation plus losartan. And we saw some interesting trends. So the classical program, so these are at the patient level, their malignant cells have been scored for each of these 7 malignant programs I showed you 2 slides ago. And you can see that the more treatment that's given, the less the residual disease expresses the classical program, and that is pretty expected. Classical program tends to show greatest sensitivity to cytotoxic therapy. And then I'll just focus you in here on the NERP program, which we didn't know anything about before this. There you see the opposite trend. There's an increase in expression of that program the more treatment you give. So since this wasn't matched data, we also took some matched organoids from some patients, untreated patients that we were able to generate organoids from. And we treated those ex vivo with chemotherapy and radiotherapy. The radiotherapy was kind of limited. We just did 5 grade times 2. But nevertheless, when we performed single nucleus RNA sequencing on these matched organoids, we also saw a significant enrichment in the NERP score. Then we went to 2 orthogonal data sets. This is bulk RNA-seq data from TCGA and ICGC. And we did a multivariable Cox regression analysis for various clinical parameters. I'm showing you time to progression. The overall survival one shows very similar results as well. And we see, again, a nice sanity check. Classical program of the 7 malignant programs is the most favorable from a prognosis standpoint. And interestingly, the NERP program is the worst. So then we wanted to look at other cancer types. So, we think this might be a little bit related to the castrate-resistant kind of neuroendocrine transdifferentiated prostate cancer. In bladder cancer, which is a little bit less well known, I found this from the TCGA study published by Robertson et al. In 2017. When they did bulk RNA sequencing, they were able to subtype at the mRNA level their tumors into luminal, 3 different subtypes of luminal, basal, squamous, and then 5% they called neuronal. And what's interesting is on their multivariable analysis, Cox regression analysis, they saw that having an mRNA subtype of neuronal had as bad of a prognosis essentially as having stage 4 metastatic disease, because they had a mixed population of locally advanced and metastatic disease. So these results, while descriptive at the human level, really made us think that perhaps we were onto something that was important in pancreatic cancer from a therapeutic resistance standpoint, and we wanted to explore it more. So at the time, I was a fellow in Aviv Regev and Tyler Jackson's lab. We had the opportunity, we were beta testing, I don't know how many of you guys have used the NanoString Genomics platform. Back then, this is my technician, she's an MD PhD student now, but it was definitely not the greatest experience back then. It's a lot better now. I think she pulled 8 all-nighters because of this instrument breaking. But those early days were definitely fun to troubleshoot. But it did give us a lot of very useful information. And basically, for those of you who are not familiar, I essentially think of this technique as, you know, an optical laser microdissection type approach. So it's not single cell resolution. We were able to get whole transcriptome as well as some proteome information, which I won't show, the proteomic side of things. But essentially, you take your molecular affinity probes, and they're all barcoded using a UV photocleavable linker. So that's kind of the secret sauce of the technique. And then you can do up to 4-color immunofluorescence. So that's how you define your morphology, your cell types of interest, right? So you hybridize everything onto your FFPE or fresh frozen tissue slice. And then you have to, this is where you need some pathology background yourself or a collaborator, ideally. You have to choose these ROIs. You're actually going to end up paying per ROI, essentially, so the cost is not just in time, it also depends on how many areas you collect. So you can do a circular region, you can do a polygon, you can do any shape. So you choose those ROIs, then you basically come in and there's this like digital micromirror device that will create a mask that will, ideally, there's some scatter of course, hit your cells of interest and only those cells of interest. It will UV photocleave those barcodes. Those barcodes get sucked up by this fax-like straw, and then it gets deposited into a 96-well plate that you can barcode spatially so that you can map it back to your ROI. And then anything higher than about 800 plex you have to read out with NGS. Otherwise, you can use this nCounter platform that basically reads out these 6 mirrors of fluorophores. So in the early days, this was our strategy. I'll show you later that we've done some more sophisticated things now with this technology. But we had ample tissue, so we serially sectioned. I'm mainly going to focus on the whole transcriptome section here, but we did an H&E, we did a protein section, and then the 2 deepest sections we did RNA. And you can see what the kind of matched ROIs look like in an example. So here is zoomed in on one ROI. So we were very interested, at the time, most people were doing 2 different segments per ROI, usually cancer and then a stroma. We tried to push it a little bit, which led to some analysis challenges by taking cancer in green, but then the stroma, we tried to separate between immune and smooth muscle actin expressing fibroblasts. And you can see with how interdigitated everything is, the fact that light scatters, you can imagine there's a lot of, you have to think a lot about the mixing of signal potentially and the order you collect things. So this is what the optical mask ends up looking like. And then I'm just going to show you some of the kind of findings that I think will become interesting in later parts of the talk. But of course, you can do a lot with this data. One of the things we did was we just did some unsupervised clustering at the ROI level. For all the malignant compartments, we scored them for our malignant programs. For all the fibroblast segments, we scored them. I didn't have time to show you, but we did a similar consensus NMF analysis and scored them for the fibroblast programs. And then for the CD45 positive compartment, we deconvolved using our single nucleus RNA sequencing immune cell type signatures. We deconvolved the ROI immune segments for various immune cell types. And we ended up getting kind of the most consistent clustering that we got. The most robust clustering was this 3-cluster solution. And we noticed something that was kind of interesting. I won't belabor this point for the sake of time. But we saw an association with this so-called treatment-enriched program, where you see mesenchymal, like an EMT subtype, which is known to be enriched post-treatment as well. And then our NERP and the closely related neuroendocrine program were associated with CD8 T cells. And we saw this when we did a different, an orthogonal analysis as well. So we found this to be very intriguing, and I'll come back to this point. And this is currently one of the fellows in my lab. His project is trying to figure this out, and whether or not the different malignant subtypes utilize different mechanisms of immune evasion. So this is new data now. So along with the Ting lab and some of our collaborators at NanoString, we did some early access work starting in 2020 on, at the time, kind of the alpha spatial molecular imager, which is now the COSMIC system. And the technology is not that different than, you know, MERFISH, for example. There are some key differences, or even the Xenium from 10x, which obviously uses rolling circle amplification, which is a big difference. But overall, these imagers, right now, we're not at the same plex as the profilers like Visium or Geomics, for example. But what you end up doing is these cycles, you hybridize your RNA-ish probes as normal, and then you do these cycles of reading out various fluorophore tagged reporter probes, and then through various cycles and basically localizing kind of like the different patterns of fluorophores, you are able to read out a barcode, right? So right now, kind of the state of the art is somewhere in the high hundreds to thousands in terms of plex. So here's, again, we have matched data across the geomics and the SNP-seq data. So again, pancreatic cancer, you're looking at some of our tissue here. And here's an example of a field of view. This was us applying roughly 1,000 plex panel. We now have 6,000 plex data as well. Not fully analyzed yet, but definitely looks more sensitive and specific than I anticipated. This image here for the field of view is protein immunofluorescence. So you can see, again, green is cancer, blue is DAPI, and then we've got a couple immune markers in red and yellow. This is the same field of view, but now it's got superimposed RNA level data. So the spatial resolution for each RNA transcript is quoted at around 50 nanometers. And you can see that by pseudocoloring based on expected cell type enrichment, so green for epithelial, blue for CAF, pink for immune, so it doesn't perfectly match the colors for the protein, I apologize. You can overall make out that the stromal epithelial compartmentalization at the RNA level is pretty good. And then zooming in to a smaller field of view within this larger field of view, you can see how the segmentation performs, and again, cell type specific transcript localization. So in collaboration with the Hamburg lab over at the Evergrande, we ran 15 samples. You can see kind of the subset of treated, which is denoted by T, U is untreated. And here is a dot plot showing various marker genes that are in this 1,000 plex panel. We were able to customize another 30 spike, a set of 30 spike-in probes as well to further be able to delineate our malignant programs. But even at this basic level of separating various cell types, immune, stromal, malignant CAF, you can see from this dot plot, it's pretty good in terms of specificity. Here's a UMAP, just putting all these cells. It's about, I think here I'm showing you about 700,000 cells. We now have about double this amount of data. But this is all obviously just in gene expression space and not actual spatially resolved information in that plot. And then for the malignant cells, again, we added some marker genes with our spike-in probes. We're able to do a pretty good job, as you can see again from this dot plot, of separating at least classical from basal from NERP cancer cells. Less good of a job with some of those other more minor programs, if you will. Like it's harder with this 1,000 gene mostly non-custom panel to separate basal from squamous, for example, or squamoid. We're able to separate the tubicin MyCap and ICAP signatures quite well, so we're pretty happy with this as our level of annotation to move forward with. So again, just as an example, I come back to that finding where this was done in kind of spatial pseudobulk resolution. We were curious, like, is this finding real, before we pursued a bunch of functional experiments and other experiments to tease out potential mechanisms of immune evasion. So at single cell level, with the spatial data, what we're able to do, and of course you can do this with any kind of single cell resolution platform, it's not unique to CosMx. We took a field of view and we just labeled the CD8 T cells in red, and the malignant cells, if they're predominantly classical, blue, basal, orange, NERP, green. And then we basically just modeled the CD8 T cells with this Gaussian decay function. We varied the decay radius, and the results I'm showing you here are robust. to decay radii from as small as 20 microns all the way to 200 microns and beyond. And you can see in this kind of topology map that where the concentration of CD8 T cells are and where the malignant cells map to it. If you do this across, I think we had like almost 200 different fields of view across these samples, and then we compare it to the null distribution, which is essentially keeping all the malignant cells and the proportion of these programs the same, but scrambling what the labels are for the malignant cells, and then doing a full change of the actual distribution with the sum of all the kind of the Gaussian decay contributions for each CD8 T cell, and then take that over the null distribution. We saw a greater than expected enrichment of NERP cells near CD8 T cells, essentially on expectation for basal, and then a depletion of classical malignant cells around CD8 T cells, very much mirroring our geomics results, which was neat to see. So as I mentioned, Ryan, who was on the previous slide, is now working on various functional co-culture and in vivo experiments to really tease out how we think basal, classical, and NERP malignant cells in pancreatic cancer interact with the immune system and evade the immune system in their unique ways. So again, in collaboration with the Hamburg lab, we were interested in improving upon some of the ways of inferring intercellular communication out there. There have been a couple other methods, from cell chat to other methods that variably looked at gene expression of ligand receptor pairs. Some included spatial information, some did not. We definitely wanted to include both at the single cell resolution. So essentially what we did was we used dbScan to generate spatial clusters of source and target cells. So we're looking at this in a pairwise fashion. And then we restricted our search space for potential interactions to adjacent clusters. So not necessarily super close to each other, but generally within a few hundred microns, where there would be we thought a higher concentration of even paracrine interactions there for secreted ligands. And then we used an optimal transport based method to assign spatially constrained cell-cell interaction likelihoods, as you can see in this table. And one of the questions that we're very interested in is what interactions can we use this method to generate predictions of that might be mediating resistance to cytotoxic therapy. So this is just two ways of showing the same data. We compared our treated samples to our untreated. In this core diagram, the thing that really jumped out to us was that CAF cells as the source and malignant cells as the target, those interactions were most enriched in the post-treated samples. Again, these are not matched samples. And then if you look here at the sum of the interaction strengths for treated in blue versus untreated in green, again, the number one most enriched category was CAF producing the ligand, malignant producing the receptor. And you can see up here, these are treated samples above the dotted line, non-treated samples below the dotted line. So we zoomed in on these CAF malignant interactions. And if you look at essentially this volcano plot, doing a DE effectively of these various interactions, you can see a number of them are enriched statistically significantly so in the treated samples, and a number of them are depleted. This was very interesting to see. On the depleted side, I'll just make a comment. You see a bunch of Wnt ligands on the CAF side and their respective receptors on the malignant side. And this is a phenomenon that we've been exploring and have seen where malignant cells, especially treatment-resistant malignant cells, will start to secrete high levels of their own Wnt, and they become niche-independent and less dependent on CAFs for producing the Wnt that they need. So it was really interesting to see that recapitulated here. On the enriched side, there were a number of hits that were interesting, and to try to narrow down things that we wanted to explore further, we did a few comparisons. So the first is we compared it to our non-spatial SNUG-seq data, and you can see here that I'll just highlight this one hit. This is CLCF1 is an IL-6 family cytokine. That hit was strongly correlated in the positive direction. It's also one of the stronger hits from the spatial data. We also wanted to go into a system that we could do various genetic perturbations in. So we took KP, the line of tumoroids, and this is ex vivo Cre recombinase. And then we co-cultured them with pancreatic stellate cells, and we generated these kind of malignant CAF tumoroids. We treated some and we didn't treat the others. We performed SNUG-seq on those. and this is the UMAP of that. And then we looked at, again, overall, not a very strong correlation, a very different model than our human data. But what was interesting is some of our hits, like CLCF1, CNTFR, again, very strongly correlated, relatively speaking, between the co-culture and the human spatial data. So we looked in the literature for this hit, and from TCGA, we see for pancreatic cancer specifically, having above median level of expression from bulk RNA-seq of CLCF1 leads to significantly worse survival. We also saw this interesting association between higher expression of this ligand, this IL-6 family ligand, with mutated KRAS status. And we found this paper from UCSF. This PI actually, we didn't overlap, but trained in Tyler Jackson's lab as well, and he's got a lab at UCSF. But he found in lung cancer, this is not in a treatment setting, but he found this interaction, this CLCF1-CNTFR interaction, to be quite important in mediating aggressive lung adenocarcinoma behavior and metastasis. And they started developing this decoy receptor and other strategies to try to target it. So, and then you can see here when they treat with the decoy receptor that has an FC domain added for stability, you see quite a remarkable regression in the overall tumor burden. So this is just one of many examples we're exploring, but highlighting, I think, how, again, kind of with my clinical hat, this is the type of thing that, I'm very interested in, where we start with patient data. We're in the middle of doing more functional follow-up with, CRISPR, genetic engineering, and other techniques. But of course, being able to find hits that hopefully have a higher rate of success in actually predicting real biology in the humans, because we started from, broad human data, validating and understanding the mechanism better with mechanistic models, and then eventually coming back to the humans and hopefully helping improve our patient outcomes. All right, so I've got about, am I supposed to stop at, what time am I supposed to stop?
Maybe 10 minutes before.
10 minutes before? Okay. This part is, I'm gonna briefly talk about, because I want to get to the cancer neuroscience part. So this is work that has been largely done by an MD PhD student, Jimmy, working with me, with Andy Aguirre at the Farber. So I'm going to go through this pretty quickly, because really what his main thesis project has been looking at is dissecting that NERP program that we described with that very descriptive data. So I'm happy to take questions later, but essentially I'm going to hit the highlights of what he found, and then spend a little bit of time on the cancer neuroscience. I know I've been talking to Dylan that was a potential interest of this group. So first thing that Jimmy did was he took other lines, metastatic, these are all human lines from the Farber, scored NERP expression before and after treatment. So these are actual match samples, and he did see enrichment there. He also took some other matched organoid samples derived from patients and showed that the NERP enrichment was unique. The classical was significantly depleted. We didn't actually see this with the basal-like program. And then looking at marker genes, I'll get to GLIS3 in a moment, but DCDC2A is a pretty good marker gene that we found for the NERP program. Again, in a matched setting in mouse cell lines, you see enrichment of these marker genes post-treatment. He also looked at other settings, so TCGA for primary, PANC-seq for metastatic samples, and what he noticed was that in primary tumors, treated or not, as expected, the prevalence from highest to lowest goes classical basal NERP, but even if you look at metastatic samples and analyze it with our programs, there's a much higher proportion of basal and NERP already, and if you look at the treated metastatic samples, you get an even stronger enrichment of these two. So really kind of suggesting that NERP is enriched in both treated and metastatic disease, essentially the most aggressive forms of pancreatic cancer. And this is just looking at CCLE at the Broad and scoring, doing a neuro-like score for cell lines representing each of these different primaries. And it's interesting that pancreas is actually just kind of in the middle, and bladder is here with the neuronal program I mentioned before. Where's prostate? Prostate's over here. So there's a possibility that a NERP-like state might be a relevant cell state even outside of PDAC that is worth exploring more. All right, so he did various computational analyses, scenic Regulon analysis, really trying to correlate transcription factors to this neural-like state and identify GLIS3 as the top candidate from various analyses. We then used DCAS9 BPR, KP, mouse organoid system to overexpress we did more than this, but I guess the highlights here are looking at GLIS3 as the putative or candidate TF for the NERP program. TRIC63, the delta N version specifically, is well known to drive the basal-like state, GATA6, the classical. And you can see the overall signatures are well recapitulated in this CRISPR activation model. So now we're able to do some functional characterization. And so Jennifer, a talented tech working with me, we did orthotopic transplants, and we took them out at 5 weeks, 8 weeks, 11 weeks, comparing a scramble guide control versus GATA6, TRIP63, and GLIS3. And you can see that at most time points, especially at 11 weeks, both the basal or mesenchymal-like state and GLIS3 have much larger tumors. And they also metastasize more frequently than the control. GLIS3 actually was the highest. Over 90% actually went to the lung. Excuse me, which was an interesting finding. So definitely recapitulate the aggressive behavior we saw. When we took these CRISPR-activated murine lines and treated them with chemotherapy or vehicle, we saw an enrichment. In this case, we actually saw a greater enrichment in the TRIC63 line than GLIS3, but definitely the biggest finding was the expected contraction of the classical line. So the question is, can we leverage some of these lines to predict other therapies? So for this part, I'm just going to leave you with this thought, which isn't fully worked out yet. But from the TCGA data, when you look at mutated tumors versus non-mutated tumors and look at their score for the neural-like state, KRAS actually scores as being the wild-type tumors that tend to have more of a neural-like state, suggesting, so Jimmy came up with the hypothesis from this, that maybe the NERP state is somewhat KRAS independent. So some other evidence of this, we got the Mirati compound that targets KRAS G12D, and this is in vivo data. We measure not orthotopic actually, this is sub-Q, but we basically treated a control versus the GLIS3 line transplanted with Mirati compound, you can see here, for over a couple months. And the NERP line definitely grows through the Mirati compound much more than the control line. If you look at organoids derived from various patients, and these are all KRAS mutated, you see actually the greater dependency you see on KRAS, the less neural-like it is. And then you can do this in cell lines as well. And this is kind of hot off the press. So I don't even have all this data yet because I need to catch up with Jimmy a little bit, but he's been working with the DepMap project over at the Broad, doing CRISPR screen on over 10 human PDAC organoid lines and looking at the organoid dependencies. And so to the right here, this is more correlated, less dependent on the neural-like state, and then to the left, more dependent on the neural-like state. You can actually see KRAS sits here on the less dependent, and you get some interesting hits, including Rho GTPases and some neurotrophins seem to be particular dependencies of the NERP-like state. So right now, along with Andy Aguirre's lab, who's a medical oncology physician scientist at the at Dana-Farber, we're very interested in exploring some of these potentially unique vulnerabilities of this NERP-like state. Okay, so all right, I'm going to skip this. This is just a summary. All right, so I'm going to spend the last 8 minutes or so talking about this interest of mine that really developed when I started as a medical student. The first pancreatic cancer patient I took care of, it was particularly horrific in a way. I mean, as a medical student, you can already basically, if you feel like you can't really do anything to help, you're there to like take notes and spend time and transfer messages to the team. But this patient was the first cancer patient I had. I've had many since, where we literally could not titrate pain control and consciousness. So basically the only time the patient was comfortable was when he was unconscious. And it was so bad, you know, especially as a new medical student, seeing this, that during one of my breaks, I wandered down to pathology, and that's when I met one of my longstanding collaborators now, Dr. Mari Mino, and I told her about this situation. She was like, do you know about perineural invasion, pancreatic cancer? It's very prevalent. And I said, no. So she showed me under a microscope, and I apologize, I don't think I have the image in this slide deck, but I still have the image that she showed me. It was just this very seared into my memory, because I remember it just looking quite grotesque. You know, you see these cancer cells that are splaying apart these nerve fibers, and that became a visual for my patient's pain. And so I continued with my medical training, but never really forgot about that situation. So when I joined Tyler Jacks's lab, I started digging. You know, I was working on other projects, like the things I showed you. I was doing some immune-related projects, but I didn't know about the cancer neuroscience field. I just started digging about peripheral tumor innervation, tropism between cancer cells and nerves, and I came across this paper. So this guy has some Boston roots. He wrote this when he was in Hopkins, but it turns out he did some work at MIT, and there's actually a graduate student fellowship named after him there. But he made a very prescient remark in 1897. He was looking histologically at a bunch of peripheral tumors, and he pondered. He said, "There's such an abundance of nerves. I wonder if these nerves are just bystanders witnessing a malignant process happening all around them, or perhaps they play a much more integral role." And you know, for good reasons, I think the cancer biology field really focused on a lot of other aspects of cancer biology in the intervening decades. And it wasn't really until the early 2000s, Gustavo Ayala and some colleagues did some seminal studies with prostate cancer cells and dorsal root ganglia sensory neurons, really illustrating. These are some representative images from some of their papers in early 2000s, showing this remarkable tropism and these contexts that these cancer cells and nerves form. This is like essentially like a scratch assay. They have a DRG with neurons growing out and prostate cancer cells growing quickly at them, proliferating more. So my interest became renewed because I was like, wow, this field really could benefit from some of these approaches that we were interested in, biological questions. So I rounded up my team. I got them excited about it. This is a clinical fellow, and you know Jimmy, and then these are two of my technicians, and there's Mari. We got together and we basically reviewed the literature, and we took a therapeutic angle. So we published this in Lancet Oncology last year, where we kind of looked at some of the mechanisms that were known. A lot of what was known was really derived from central nervous system tumors. So Michel Mange and Frank Winkler and others have done a lot of great work looking at those interactions. And what I was kind of found remarkable was that, you know, I knew about the morbidity, right? I knew about neuropathic pain, and there's some mechanisms that may explain it. I was floored when I saw this morbidity, because as a clinician, I was always taught that patients with brain tumors, they're at risk for seizures because of mass effect, right? The skull's not going anywhere. If the tumor grows, you can herniate, you can induce hyperactivity, seizures. So we always, you know, prophylax them with Keppra or whatever your AED of choice is, and then give Ativan if they start developing signs of seizures. But it turns out that Michelle Mange and her team showed that there's really this very intricate mechanism where neurons in the brain can actually synapse onto these glioma cells, secrete neuroligin 3, which gets cleaved by this ADAM10. That binds to a neurexin receptor on the postsynaptic cell. Huh, interesting. Wow, I just found an error in this image. This is mislabeled. All right. Well, anyways, this is flipped. But anyways, the cancer cell, it basically has PI3 kinase mTOR signaling stimulated, and then here's the cool part: it feeds back onto the presynaptic neuron and secretes a ton of glutamine, and actually lowers the excitatory threshold and stimulates that neuron to actually fire more and produce more neuroligin 3 in this positive feedback loop. And that is at least one of the mechanisms by which brain cancer patients are more susceptible to seizures. So I thought that was pretty cool. And then there's other things that I think are you know, maybe still unconventional to think about, but you know, promoting tumor development and growth, stress adaptation, including treatment-resistant metabolic stress. Alec Kimmelman had a beautiful paper in Cell a couple years ago looking at what happens when serine-starved cancer cells are in that type of depleted microbiome. They'll actually selectively translate NGF to bring nerves to their local microniche to secrete serine to them. Metastasis, immune remodeling. So really interesting. All right, but you know, while we know a little bit, this is a summary from Lei Zhang's lab of at least the biochemical communication. We're starting to learn about immune modulation. Peripherally, we don't know very much about metabolic support and even electrical communication. So my team was kind of interested in this gap that we saw, where if you look at all the prior studies in the cancer neuroscience field, especially peripheral studies, they generally focused in on a hypothesis, a pathway of interest, and then they also lacked cell type specificity and spatial context, which is really important because you know, when we look at a perineural invasion positive tumor, it could just be one focus, right, on the entire tumor. So if you're averaging that signal and trying to understand what allows cancer cells and neurons to communicate with each other, you're going to be lost in a bunch of noise, we thought, right? So this is just an image of perineural invasion. So real quick, is it okay if I go a few more minutes, or? Sorry, I tend to be long-winded. So during the pandemic, Jimmy, my tech Hannah, we were a team of 3 at the time. We had a combined 13 hours in the lab, so we were basically just keeping up our mouse colony. So we decided to hone our in silico skills a little bit. So I had noticed as a resident that when you go into like cBioportal, which houses the TCGA data, that there's a slide you can click that's like histology or pathology, and you actually get the redacted pathology report, and then you get the H&E images. And I always wondered, you know, I've seen a lot of TCGA-based studies, but I hadn't seen any studies that really utilize that information. So we were bored, so they went through across 10 different cancer types in TCGA that had bulk RNA-seq data, clinical annotation, the pathology report, and H&E images. They went through and wrote down every patient that had lymphovascular invasion, perineural invasion, or not. So we had like over 2,000 patients. And the first thing we did in this paper was we just looked at enriched in perineural invasion versus not. And we looked at, from the bulk RNA-seq data, what was enriched. And this was a good sanity check for us. We saw some neurotrophins like somathrins, GDNF. We also saw a bunch of EMT-related genes with MENTA and MET. That will come back later. But this project we had fun with because we essentially ran through a bunch of ML algorithms, did a grid search. Random Forest ended up winning. But we were able to use kind of bulk RNA-seq data and generate a signature that we hoped could predict occult PNI, because believe it or not, if you look at some of these cases, these were all missed by the initial TCGA pathologist. There's a mask on here on the H&E. But you can see these, once you see them, you can't unsee them, right? These are cancer cells in this light green, purple nerves. And we basically did a power calculation, gave a different pathology collaborator a bunch of TCGA slides and said, hey, can you tell us if these have PNI or not? She didn't know what the classifier score was. It turns out half of them were high, half of them were low. And she also didn't know what the TCGA pathologist called. And if you look, she overturned from negative to positive for PNI significantly more in the high classifier group than the low classifier group. So essentially supporting that, you know, we were able to detect occult PNI with some confidence. Okay, so in the last couple minutes, I'm just going to give you a little teaser about what we've been doing since. So I wanted to address those limitations that I showed you before. So we started with geomics because it actually is quite well suited for this. It's whole transcriptome. We don't have to be biased to a particular set of candidates. But we were able to, we went beyond what the kind of onboard software allows us to do by doing the following. So it took about a year to generate. We generated 288 cores like this of matched regions in pancreatic cancer patients with nerve involvement, no nerve involvement but cancer, and then areas with nerves but no cancer. And we arrayed them here. We did whole transcriptome DSP, and you can see this is kind of the overall, the number of ROIs, AOIs, and we did an inch, so this is all the TMAs. And we did a kind of, I think, an interesting segmentation strategy. So here's an example, ROI. So we just put together a pretty simple custom script in ImageJ that would allow us to, within the ROI, first sample the nerve transcriptome, then the cancer cells within the 100 microns of the nerve, and then cancer cells within 300 microns, and then the same for fibroblasts. We can do this for immune cells too. So we get up to 5 segments from every ROI. And you'll recall these subtypes, right? So we did some simple analyses initially. So when you look at segments, malignant segments in areas with PNI versus no PNI, we saw a very strong enrichment for the mesenchymal program, kind of recapitulating the EMT genes we saw in our bulk analysis. We saw a smaller but significant enrichment in the nerve program, a depletion in the classical program. I have to split, yeah. When we split this into large and small nerves, this was quite interesting because with the large nerves, this is kind of an example. These are probably intrinsic ganglia that were already there before the tumor developed and they invaded. We see the mesenchymal and the classical, the basal is related to mesenchymal, so they always seem to go hand in hand. We saw those associations, but the nerve program had no association. But we only saw the association when we looked at the small nerves. So everything is recapitulated from the mesenchymal and basal, but the nerve program was only associated with the presence of these small nerves, which kind of suggests more of a neo-innervation pattern. So we came up with this hypothesis that EMT, so classical cells are excluded from nerve-infiltrated areas. That seems like a pretty clear finding. But we had this hypothesis that perhaps EMT programs facilitate nerve invasion regardless of nerve caliber, while the nerve program may be more subtly associated only with small nerves and not large nerves because perhaps they play a greater role in mediating nerve recruitment and outgrowth rather than invasion. So we're testing this hypothesis in various different ways. This is at the gene level, a DE analysis, a spatial DE analysis. There's colored by what program those genes are strongly weighted in. This is looking at gradient information where blue are our positive hits, so more enriched as you move towards the nerve. Red is one of our example anti-correlated hits. And you can see overall, other than one exception, as you move closer to the nerve, you see this kind of monotonic increase in gene expression. We've gone into our CRISPR activation model to overexpress. We're also doing knockout of these various candidates. And we're looking at, for example, invasion. So this is kind of invasion through matrix gel, comparing candidates to a scramble guide. I'll show you this video because I think it's kind of cool. This is a 3D invasion assay where we've got a dorsal root ganglia in the middle, and these are TD tomato positive cancer spheroids that are invading through matrix gel. And we can look as a function of genetic perturbation, the kinetics, you'll see this kind of tropism. These organoids or tumoroids actually migrating towards and actually invading into that dorsal root ganglia. And if you take that same image and you just do some high resolution confocal, we're noticing that these little tumoroid structures actually adhere, seem to be tethered by these nerve fibers that are emanating from the DRG and actually track along them. And we pluck the DRG out here and image it separately. You see these TD tomato positive red cancer cells have actually invaded into this ganglia. So Peter, C32 postdoc in my lab, and the technician working with Nicole are doing some really cool experiments here. This is to show you what other functional follow we're doing. These are dissociated neurons. We can use conditioned media experiments. We can spike in exogenous protein and look at outgrowth kinetics. So I'll stop there. There's a lot of future directions that we're interested in, and certainly many that we don't have time to do right now. And one of the areas that I put on my first slide is, so this is Tyler, if you guys don't know, my postdoc mentor. We recently were excited that they chose one of these co-culture images where you see cancer organoids and these nerves forming these dense networks among the various organoids in the Koch gallery over at MIT. So that'll be up for like a year, so that was pretty exciting to see. So with that, thank you guys very much for your attention. Thank everybody who is part of the team, my clinical collaborators, my postdoc mentors, Tyler and Aviv, and all of our funding sources, our patients and their families, and then all of you for your attention and this kind invitation. Thank you. Yeah.
Great talk, thank you. I have two questions. When you look at the model, I know you mentioned the state of T cells. What about
Yeah, great question. So cursory level, I would say. So one of the new postdocs is actually trained as a macrophage biologist at WashU and is looking into more questions there. But that is definitely an area that we have not fully tapped into in our data set yet.
The second question is, do you think the nerve is talking directly to the cell, or like speaking it by something components, and then by priming like the environment and then something?
I think probably a combination. I think, Uli, Von Adrian, and others have, not in the cancer context necessarily, have really looked at like neuro input to like lymph nodes and reprogramming there. I think that will certainly play a role in anti-tumor immunity. I also think I was gonna see if I could pull up some interesting images. I'll give another similar talk as you guys can see. Let's see if did I not put this here? we're starting to see some very intimate interactions between the cancer cells and the neurons directly. Let me see if I can show you this image. Well, so this is very preliminary, but oh shoot, sorry. You guys have like no idea what I'm talking about. Maybe that was the case earlier too, but now it's especially challenging. Okay, so this is very preliminary. That's why I even hesitate to put it on. But we've done some preliminary even synaptic staining, and this is not the best images. This is just what I can find right now. But we definitely see like a concentration of. So these are some classic presynaptic, postsynaptic stains. In areas where there are nerves and cancer cells, cancer tumoroids, we see much higher concentrations of these, and some of them at high res, we haven't done electron microscopy yet, but some of them definitely look like it's a cancer cell on the postsynaptic side, but it's not definitive yet. So to answer your question, I think it's going to be a combination. I don't think it's going to be exactly the same as what they're seeing in the central nervous system, which is pretty nuts. They've actually found pacemaker cancer cells that express a particular voltage-gated potassium channel that it's like that you also see in the SA node of the heart, and that if you ablate those, another cancer cell nearby will undergo plasticity and take over that role, and they communicate, you know, calcium signaling amongst them through connexin-based gap junctions and things like that. And Frank Winkler just published a Nature paper on that. Pretty cool. I don't know that it's going to be to that extent peripherally, but I would not be surprised if there was some electrical as well as biochemical and metabolic immune-mediated communication peripherally as well.
I have one question on your first part. So you showed that IL-6 is accumulated in the region like where there is like a lot of nerve growth in the tumors which are resistant. So what do you think, like what would be the role of IL-6 or in the growth of nerve cells or the tumor cells or like the adjacent cells? And what induces the growth of like nerve cells in the tumor?
Yeah, so good question. I'm trying to, my exact comment I think was with IL-6 related was this CLCF1, which is in the family, and you can see from this diagram that, you know, on the receptor side, you can see why it's part of the IL-6 family. So I think there's a couple interesting things to say from your comment. One is that IL-6 is one of the markers that we see in inflammatory CAPs, which are associated with bad prognosis, actually in many cases worse than the MyCAPs, that are the myofibroblastic CAPs that just create this dense stroma and things that we traditionally associate with bad outcomes. This makes me think, right, because we're seeing this strongly enriched in three different, two ways of looking at human data, one spatial, one not, and then a model system that's very different. And this was the #1 kind of concordant hit that when you treat with cytotoxic therapy, this signaling increases. So I do think that there's the potential that IL-6 and IL-6 related cytokines are directly signaling and, you know, increasing. We know it acts on you know, JAK-STAT3 signaling and other pathways downstream that would promote tumor survival and proliferation. And so I do think that could be one of the mechanisms for worse outcomes, and ties together why the prevalence of ICAS producing IL-6, and then we're finding that CAFs in the post-treatment setting producing this IL-6 related cytokine, you know, might actually all tie together for worse outcomes. Not sure about the nerve stuff yet. This part was not as related to the nerves yet.
The great talk, I really enjoyed it. The connection between the pancreatic cells and the nervous system is obviously really intriguing. And I'm wondering, do you think this is a mechanism that sort of happens at some other time normally? Cancer cells are reactivating it, or somehow have the cancer cells sort of creating this new process? What is it in cancer cells?
Yeah, no, I think that's a fascinating dichotomy to think about. It's unsatisfying, but I suspect the answer is probably a combination. But to the first point, I think, so this was actually a while ago. First author was Magnon, published in Science probably around 2015 or so. This really beautiful study where they had double cordon positive neuroprogenitor cells from the subventricular zone of the brain, and they were able to trace, this was in a mouse model, but they were able to trace that those cells were able to find their way into the prostate bed before a tumor had formed and differentiate into more sympathetic like neurons, and that it was important that they were sympathetic like, and that kind of beta adrenergic and other signaling fostered initial development in this model system of prostate adenocarcinoma. But that later they transdifferentiated, this was also important for dissemination, into more of a cholinergic phenotype, and that if you kind of ablate that or block that signaling, then you don't get as much metastatic dissemination. I suspect that it's kind of like nerves develop very similar to the vascular system, right? They're always in parallel, right? And we've studied that a lot more. I think mostly the cancer cells are reactivating some of these like developmental pathways where the nervous system is guiding, providing cues for physiologic tissues and structures and polarization and all those things. And I think in some ways, the nerve program, we called it neuro-like progenitor because we were seeing some of that stem-like stuff and the hepatocyte nuclear the factors that were also enriched. I think it's also a state in which there is greater plasticity. And also, I think about, I don't have definitive evidence for this, but I think about how resistant nerves are sometimes to cytotoxic therapy. Sure, sometimes we get peripheral neuropathies from oxali, but like from the radiation standpoint, we can give like 90 gray to, for trigeminal neuralgia, right? You don't want to treat that surgically, right? They're going to, that's facial nerve, things like that. You can give incredibly high doses of radiation, and the nerve is for the most part fine. They get a little demyelinated temporarily, things like that, but they preserve function, and that's way high enough to eradicate the cancer for the most part on them, at least transiently. So there may be some inherent nature of being more neuro-like and resistant to cytotoxic therapy as well. So I think there's probably multiple advantages. Yeah.
Really amazing talk. Maybe my first question follows up with Mark's question. If I understand correctly, so this neural progenitor-like cells, is it purely intrinsic, or might there also be some chromosome rearrangements that might potentially increase some kind of like upstream pressure factor that drives that, and that's why you also see it in other types of cancers?
Yeah, that's a great question. Something that we didn't explore in that paper at all, but actually right now there's an ongoing study where there's multiple ways to probably do this, but we're generating some single cell whole genome as well as full transcriptomic data from the same cells. It's a costly, this is at the SNP level, right? So a lot of the GNT-seq approaches before, you could really only get like CNA level information. So we're generating that data now, and I think that's going to really go a long way in kind of answering that question. But even at the CNV level, you can answer, broadly what you were saying. I suspect that you're right. I think there's going to be some common themes there, and there's probably going to be a whole layer of epigenetic modifications that ultimately manifest in this state that has some commonality in different contexts, as well as different tumor histologies.
So for the ligand receptor nodes, you basically showed here, what I've seen previously, and you have the data to check it, is that a lot of the ligand receptors oftentimes are only a subset of the cells. So basically they just subset these cells you generally see. Do you see that in your Cosmix data set? Or is it more like evenly? Is it more like, is there variability at the single cell level?
I actually haven't, I just for the sake of time, I didn't put the image in, but I can share it with you later. We have, for all the FOVs, the treated and untreated, we actually have it mapped out. Spatially, we have it superimposed with, one color for the receptor, one color for the ligand, and the size of the dot is essentially the expression level, and you can see where they're spatially located. Interestingly, for this one, CLCF1, CNTFR, it's not true for all the hits. This one is actually pretty even. I have not quantified it, but it's when you look at the FOVs and the treated, it's all over the FOV. It isn't isolated pockets, suggesting a particular subclone or something like that.
Maybe one more question, sir.
Yeah. So the treatment is increasing basal-like cells and the neuronal-like cells. So the treatment is enhancing the invasion of the nervous system, of the nerve system. Did you say that? So the treatment, therefore, is increasing more discomfort for the patient.
I think that's an interesting thought. I don't know if I would go quite that far. I think we're selected. There's some. So the other study we're doing is some lineage tracing studies with Hwang fellow Arnav, and looking at how much plasticity versus selection there really is. Because I mean, we're obviously inferring that with a lot of what we're doing here. I think that you're definitely not wrong. It's interesting. I've had a couple patients where the initial tumor did not have much perineural invasion. They get treated, they're doing fine. The recurrence, which presumably there was, especially a local recurrence, there was probably some, microscopic disease there that we didn't know was there. When it grows back, the few cases I'm thinking of, they often are more lymphovascular, perineural, invasive. I don't know if we can't go as far as saying the treatment is causing that to be the case. I think the cells that survive the treatment may be the ones that are more, and they're the ones that proliferate in the aftermath of treatment. All the other cell types have died, right? The resource environment is different now. But I do think that there is probably some plasticity too, in which case, yes, right? To the extent that we're taking the classical cancer cell and treating it, and then it's morphing into a NERP-like or ENT, post-ENT cell, then I think what you just said would be more accurate, right? Because that's a more aggressive cell. But the selection part, I think, is still an important part of the puzzle, where it's already there. Yeah. Cool. Thanks so much, guys.
Thank you, everyone.